Okay, here is our next lesson. We're going to talk about meiosis and how it's a source of genetic diversity. That's going to become really important when we talk about evolution in Unit 7. So just a reminder, when we have meiosis happen, that's going to separate homologous chromosomes during meiosis 1, sister chromatids during meiosis 2, and the end result is going to be producing haploid gametes, each of which is going to receive a full set of chromosomes that is going to have a mix of both maternal and paternal chromosomes. Now, this process can go wrong. You can have something called nondisjunction occur. And nondisjunction is a really complicated term for what should be a fairly simple concept. Let's break the word down. Junction means like an intersection, a joint of something. Disjunction means when that joint comes apart or separates. So normally during meiosis, you're going to have disjunction of the homologous chromosomes. They're going to separate during meiosis 1, and then you're going to have another disjunction of sister chromatids during meiosis 2. Non-disjunction means you do not split apart that joint, and so you have a failure of chromatids or chromosomes to separate. So this results in gametes that are not going to be fully, completely haploid. They are called aneuploid, meaning they don't have the correct number of chromosomes. Aneuploid could mean that they're lacking a chromosome. They could be N-1, or they could have an extra chromosome, which means they could be N+1. Now, if the meiosis event happens, if the non-disjunction event happens during meiosis 1, what will happen is homologous chromosomes don't separate, and then all of the gametes that are formed are going to be aneuploid. They'll either have one extra or one too few chromosomes. If the non-disjunction event happens during meiosis 2, then half of your gametes, the ones that are formed from the cell during meiosis 1 that correctly undergoes meiosis 2 and separates sister chromatids, those half of the gametes are going to be completely haploid and are going to be normal. The other half of the gametes are going to be aneuploid. So if you have sister chromatids fail to separate, you'll have in the end produced a gamete that's going to have two copies of the sister chromatids, so two identical chromatids, and then you'll have another gamete that's going to be lacking a chromatid. So it will have one less chromosome. This results in something called trisomy, which is where you have an extra copy of one chromosome, or monosomy, which means you have one less copy of a chromosome. We're only going to talk about trisomy, which is where you have three copies of one of your chromosomes. Literally, trisomy means three bodies. Now, most autosomal trisomies are lethal, meaning they will not even result in a live birth. The most common trisomy that exists is trisomy 21, which is Down syndrome. It's the main autosomal trisomy that can result in not only a live birth, but a person who can actually live to adulthood, depending on the severity of the trisomy. So here we see a karyotype or a picture of the chromosomes of an individual who has trisomy 21 and has Down syndrome. You can also have trisomy of the sex chromosomes. So you can have an extra X chromosome. And so if you have an extra X chromosome, you can have three X chromosomes, or you can have two X's and a Y, or you can have an extra copy of a Y chromosome, which would be XYY. Normally, you might see some small phenotypic differences, but most of the times people will not know about having this trisomy unless they have reproductive difficulties later on in life. All right, so let's talk about the major sources of genetic variation during meiosis. The biggest one is going to be crossing over, otherwise known as recombination. This happens during prophase 1. And what happens is when homologous chromosomes pair up during synapsis, when they're forming those tetrads, non-sister chromatids exchange genetic material. They can swap parts. So if we look at these two chromosomes here, the blue chromosome has all dominant alleles, big A, big B, big C. The recessive chromosome has all recessive alleles, little a, little b, little c. If no recombination happens, then you could only inherit dominant alleles or recessive alleles. However, after recombination, when you have recombinant chromosomes, then what you can have is a mix of dominant and recessive traits. That allows for greater genetic variation, a mix of maternal and paternal traits to be inherited in a single gamete. And this ultimately increases genetic variation. Now, during sexual reproduction, you actually have 3 points that are going to be major sources of genetic variation. The first one we've already talked about, which is crossing over. There's also the independent assortment of chromosomes during meiosis. So when we did our meiosis chromosomes activity during class, you saw that you could inherit a mix of maternal and paternal chromosomes. That's independent assortment. And then when fertilization of gametes happens, you also have genetic variation because you'll have a mix of different chromosomes coming in from the father and a different mix of chromosomes coming in from the mother. So if we look at this in diagram form, here's one source of genetic variation, which is crossing over. That's going to create some variation. Here is independent assortment. So notice, depending on how these chromosomes line up, or how the homologous chromosomes line up during metaphase 1, you have a different possible mix of maternal and paternal chromosomes. And then finally, during fertilization, you have different combinations of these chromosomes that can be inherited from the gametes of two different organisms, and all of that is going to lead to a dramatic increase in genetic variation during sexual reproduction. This is going to be really important when we talk about evolution because genetic variation is almost basically fuel for evolution. And you'll find that sexually reproducing organisms will have an advantage during times of environmental change because they have this increased genetic variation. All right, that's it. And our next lesson will be the final one that I'm planning to do this year for Unit 5.